Cellular Respiration and Fermentation, Chapter 9. The best way I can describe this chapter as an opener is me breathing in and then me breathing out. Breathing in and breathing out. That is cellular respiration. So the question is why are we always breathing? Do we need to breathe? And the other question is, do we always need to eat? And what's up with fermentation? So that's what this chapter is about, is how is it that we get energy to survive. Remember what energy is? It's one of the 5 characteristics of knowing that something is alive. Okay, so in this chapter, we're going to learn how cells make ATP starting from sugars and other high potential energy compounds, and how we produce cells, or ATP, in the presence of oxygen, and to know the 4 steps of cellular respiration. Then we'll also talk about fermentation, in other words, the production of energy in the absence of oxygen. So this chapter is all about producing energy. So remember, life requires energy, and it's one of the characteristics of life itself. And ATP is used to fuel that energy. So when we talk about energy, where is it that we get it from? That's why we're constantly breathing. Recall that I mentioned that ATP, there's only 2 ways that energy can move around in the cells. It either moves around with electrons or it moves around with phosphate. So ATP has 3 phosphate groups, so ATP has a lot of energy. But the thing is ATP is a very unstable molecule, so we cannot store large quantities of ATP. So we need to produce it as we need it. So that's the beauty of life. It's not too much or too little, it's just right. And this just right means that we make energy as it is being used. Now, that's a good question. What do we mean by synthesizing or making ATP? That means we need, when that one ATP is broken off and it is that energy is used for another reaction, how is it that we can reconnect it to form ATP all over again? Well, that's where the glucose comes in. We need glucose to have that energy to reconnect that phosphate group that was broken off and reconnect it to make ATP. So when you see ADP means it has adenosine diphosphate and only has 2 phosphates. So to connect that 3rd phosphate to make ATP or synthesize it, we need energy, and that energy is going to come from glucose. But where in the glucose molecule? It's going to come from the bonds, right? Isn't that where potential energy is? We break the bonds and it releases energy. Cells generally contain enough ATP to sustain from 30 seconds to a few minutes of activity. Like I mentioned before, ATP is a very unstable molecule, so we make it as we need it. And for those of you who exercise a lot, you know the term hit the wall. That is, when you run out of energy, it's because you've consumed all of the ATP, but if you stop and you rest a little bit, you get like a second wind. Well, what happens in that second wind is your body has made more ATP. It has synthesized more ATP for you to continue. So we need to eat so that we can produce our own energy. So we're going to use glucose as an example of how energy is formed in our cells. So plants produce glucose during photosynthesis, which will be our next chapter, and then we eat food so we can have glucose. So it says here organisms store glucose as glycogen or starch. So in animals it's stored as glycogen, and in plants glucose is stored as starch. So this is a classic formula, and you should know this, like backwards and forwards. So it says The breakdown of glucose in the presence of oxygen will give us carbon dioxide, water, and energy. Now notice here it says heat and light, but aren't these forms of energy? So we could put at the end of the equation ATP. So in other words, can we not say that cellular respiration is the oxidation of glucose? Glucose is oxidized. Glucose is losing electrons. Glucose is losing electrons. We're going from a 6-carbon molecule to a 1-carbon, which is CO2. So this is the oxidation of glucose. So another name for cellular respiration is the oxidation of glucose. And this is done through a series of redox reactions, oxidation and reduction. Fermentation will also oxidize glucose and form ATP, but the truth is we're going to learn that really it can only form very limited amounts of ATP. So oxygen really is the key of forming or maximizing the amount of energy that can be formed. Cellular respiration produces more energy than fermentation. Let me say that again because it could be a test question. Cellular respiration produces more energy than does fermentation. This is a great slide, figure 9.1. It shows you the combination of photosynthesis and cellular respiration and how it's used to produce energy. And here energy is needed to actually for those people that are rowing their boats. So we can make energy in the presence of oxygen, or we can make energy in the absence of oxygen. So in either case, ATP is produced. So it says here what happens when glucose is oxidized. So in cellular respiration, there are 4 steps. I love this slide. You should put like a couple of stars here to remind you that you need this slide because this lists all 4 steps. Glycolysis, pyruvate processing, the citric acid cycle, and the electron transport chain. So if you look at these 4 steps, we start off with a 6-carbon molecule glucose, and it is broken down completely by step 3, the citric acid cycle, to a 1-carbon compound, CO2. And then all of the energy in the form of electrons will be taken to step 4, the electron transport chain, and that energy is going to be used to pump protons on one side of the inner membrane of the mitochondria, and as it crosses the inner membrane, it's just going to produce gobs and gobs of ATP. And we'll go through these steps so that you can see how this works. But I do love this slide, so if I were you, I would mark it as an important slide for studying. I also love this slide too because it's a diagram that's asking you what goes in, what comes out, and where is it happening, each of the steps of cellular respiration. So we see that glycolysis, the first step, glycolysis, the breaking of sugar, happens in the cytosol. And notice glucose goes in and ATP, pyruvate, and NADH comes out. Well, really, NAD is an electron carrier, so that indicates that actually it's becoming reduced. How do I know that it's reduced? Because it has an H at the end of NAD. So I know that it's reduced and it's carrying electrons. In step 2, pyruvate processing, this happens in the mitochondria. This is happening in the matrix. And notice here, pyruvate goes in, and what comes out? CO2 and NADH. So where are the electrons coming from? It's the breaking of covalent bonds, right? Isn't it true that a covalent bond is the sharing of electrons? So when those bonds are broken, there's a release of energy in the movement of these electrons. And then in step 3, acetyl-CoA, which is a 2-carbon compound, goes in. And what comes out? NADH, FADH2, CO2, and ATP. So notice here is that we have electron carriers, two types, and then glucose is now completely broken down to CO2, and also ATP is made. So notice ATP is made directly from the substrates that are being broken down. This is called substrate level phosphorylation. So it's in step 1, glycolysis, and in step 3, the citric acid cycle. Then all of those electrons, all of those carriers go to step 4. And step 4 is in the inner membrane of the mitochondria. And notice what goes in and what goes out. So we know that electrons are going in, but notice that O2 is also going in. So what is being produced? Water and ATP. And most of the ATP is produced in step 4. I really love this diagram. What goes in, what goes out, and where is it all happening? So what happens when glucose is oxidized? When glucose is oxidized, it says here cellular respiration is a set of reactions that uses electrons from high energy molecules to make ATP. So remember I had mentioned before in chapter 8 that energy moves two ways in the cell. It moves through electrons and it moves through phosphate groups. So in cellular respiration, we're going to see that a lot of energy is moving through electrons. Energy is the source from glucose to actually generate ATP. Remember, we're reconnecting the phosphate group to ADP to produce ATP. So when we're eating things that have glucose in them, that material is used to make energy, and we also use those materials to make more of us. Okay, so remember we talked about catabolic pathways and anabolic pathways in the previous chapter. So keep in mind that a catabolic pathway means that we're going from a large molecule to a small one. In other words, we're breaking down molecules, and that energy that was that potential energy in the bond is going to be used to produce ATP. In an anabolic pathway, it's just the opposite. We're going from small molecules and we're going to use them as building blocks to produce large molecules. And this is going to require energy, and that energy is ATP. So we can put catabolic and anabolic pathways together. One releases energy and one requires energy. This is energetic coupling. Cellular respiration interacts with other pathways. So in this chapter, we're basically going to talk about the production of energy, but the truth is this four-step pathway is a miraculous pathway, and it's used for so many other things within the cell. So let's start at the very beginning. We're starting with glucose, right? But the truth is, we don't have to use glucose. We can use fats and we can use proteins. And you don't have to listen to this lecture to know this. You guys know that if you go on a diet, like if you go on a paleo diet or any kind of diet that cuts out sugar, you know that you're still going to have energy. So where does the energy come from? It comes from the fats and the proteins that you eat. It says here that fats can be broken down at different parts of this metabolic pathway that we call cellular respiration. And the same thing will go for protein. Now, I'm going to move quickly to the next slide because I just want you to know about this. We're going to use glucose as our starting point, but you intuitively know that we can use fats or proteins. So it says here that there are also intermediates in cellular respiration, and these intermediates can be used for other for other reactions within the cell. And notice I am also going to go fast through this slide too, but it doesn't take away from its importance. I'm going to show you a diagram in a little bit that shows you that we can have different starting points and we can have intermediates along the way that are used for other reactions. So this is the figure that I wanted you to see, figure 9.3. So if you look at the very middle of the figure, you see glycolysis, pyruvate processing, and the citric acid cycle. In other words, steps 1, 2, and 3. But so here are steps 1, 2, and 3. But if you look above, you can see that we start off typically with glucose or sugars for cellular respiration, but you can start off with fats or you can start off with proteins. So it doesn't really matter. So we're starting off with carbohydrates. Now, if you look below, you'll see anabolic pathways. Remember, anabolic are building pathways. So here you see that we have substrates for nucleotides, substrates to build phospholipids and fats. So here are the intermediates that can be used for other reactions in the cell. So this is a complicated pathway that for us, I want you to know the energy producing pathway for now. So remember, catabolic pathways are used to break large molecules down into smaller ones that release energy, and anabolic pathways are just the opposite. Okay, so we can organize these pathways, but they usually work coupled together, and this eventually forms a situation called homeostasis. It's like a seesaw. We keep things in balance. It's not either one or more of the other. They're working with each other. I love figure 9.4 because it shows you actually the real complexity of cellular respiration or the oxidation of glucose. It can be used for so many things. So it's just something to keep in the back of your mind. We're learning the basics of something that has many functions. I particularly like this slide because if you can answer these questions, then you know this entire chapter and you will do very well on the upcoming exam. What goes in, what comes out, what happens to the energy that is released in each step, where does each step occur, and how is it regulated? If you can answer these questions, you understand the whole chapter. So remember, use this as the questions you're going to ask for each of the steps. Okay, so let's start off with step 1, glycolysis. This is the first step into oxidizing glucose. So when I read glycolysis in my head, even though I know it's incorrect, I say glycolysis. What do I mean by that? Glyco represents glucose and lysis means to break. So the breaking of glucose. So we're starting off with a 6-carbon sugar and we're breaking it into 2 3-carbon molecules, and these molecules are called pyruvate. So they're both each 3 carbons. So this is happening in the cytosol. That's that space between the nuclear envelope and the plasma membrane. So this takes a series of steps and itself is a metabolic pathway, and the end product is 2 pyruvates. We start off with 1 glucose, 6 carbons, and we end up with 2 pyruvates that have 3 carbons. Okay, glycolysis is 10 steps. Do you need to know each of the 10 steps? And the answer is no. But do you need to know in the back of your mind that it takes 10 steps, that it's complicated and sophisticated? And the answer is yes. So I love this slide because it shows that to initiate the whole beginning of cellular respiration, you actually need 2 ATPs. And these ATPs are going to be an investment. And a total of 4 are going to be made. So we have a net gain of 2. 4 minus 2 is 2. So we needed 2, we made 4, but we have a net gain of 2. We also make 2 ATPs. ATP is made from the breaking of glucose itself. This is called substrate level phosphorylation. And of course, we also have NADH. So we have released high energy electrons that are being shuttled by these little molecular taxis, and this one is NADH. So this one is reduced. So this is also found in your book, and here you can see the energy investment at the very beginning. Here we have 2 ATPs. This is the investment. And then see that enzymes are being used to actually, this is used to phosphorylate the substrate, and it's making AP. So here are all of the 10 steps. Do you need to know these 10 steps? No. But you know what's good to know? You need to know that hexokinase, phosphoglucose isomerase, and phosphofructokinase are one of the first enzymes in this 10-step process. Because remember, we need to know how to regulate glycolysis. So remember, feedback inhibition, the products at the end affect the enzymes at the beginning. So these are the beginning enzymes. So notice here phosphofructokinase is the third enzyme, and this is, we're going to regulate glycolysis through what? Feedback inhibition. The product at the end affects one of the enzymes at the beginning, and this enzyme is phosphofructokinase. So a lot of ATPs will inhibit phosphofructokinase. It's saying stop, just stop. We don't need any more ATP. Okay, so here you see how ATP regulates actually phosphofructokinase, one of the first enzymes at the beginning of glycolysis, which is a 10-step process. So notice it's at a secondary site. So in eukaryotes, pyruvate is produced by glycolysis and transferred over to the mitochondria. That's why we say the mitochondria, its primary function is to produce energy. Okay, now if we're talking about a prokaryote, actually this process continues in the cytoplasm. So I want to remind you that the mitochondria is an organelle that has two membranes, an outer membrane and an inner membrane. So the outer membrane is the one that you see on the outside of the organelle, and the inner membrane, it folds in and out, and it forms like these little sacs called cristae. And this is important because some of the reactions are now going to occur either in the matrix, the liquid part, or they're going to occur in the inner membrane of the mitochondria. Okay, so here we go. So here you see that the purple and the blue membranes show you that there's an outer and inner membrane. And in between the inner membrane is that space called the matrix, the mitochondrial matrix. And this is important because we will have the steps, now all the remaining steps, steps 2, 3, and 4 actually occur in the mitochondria, of course in eukaryotic cells. Okay, so step 2 is pyruvate processing. And we're going to go from a 3-carbon molecule, which is pyruvate, to a 2-carbon molecule, which is acetyl-CoA. So what's happening here? This one is going to occur in the mitochondrial matrix if it's a eukaryote. And remember, in prokaryotes, there's no organelles, so it has to occur in the cytosol or cytoplasm. And then here it says, when it does, where does that CO2 go? Because remember, energy cannot be produced or created or destroyed. It has to be transferred or transformed. So that one carbon that is broken off is going to form CO2. And so the remaining 2 carbon unit is attached to coenzyme A, producing this molecule called acetyl-CoA. In figure 9.9, we see that pyruvate has 3 carbons, 1, 2, and 3. And see, one of the carbons is in red. This indicates that it will be broken off and released as CO2. So when a bond is broken, it's going to release more electrons. That's why NAD becomes reduced. How do I know it's reduced? Because there's a proton at the end. And now coenzyme A is added to the 2 carbon molecule, producing acetyl-CoA. So this is step 2, and for eukaryotes, it occurs in the mitochondrial matrix. Pyruvate processing is also regulated by feedback inhibition. So if there's too much ATP, then pyruvate processing is going to stop. It's just going to stop. Now, if there's not a lot of ATP, then it's going to say, go, let's go. Or if you have a lot of glucose, it might say, let's go. If there's a lot of pyruvate, it's going to say, let's go. So feedback inhibition is going to regulate pyruvate processing. Okay, so here it is. So we have pyruvate processing. Step 3 also occurs in the mitochondria. Step 3 is the citric acid cycle. In the citric acid cycle, each acetyl-CoA, which is a 2 carbon molecule, will actually produce, will be broken down to form 2 CO2s. So we went now from a 6 carbon glucose molecule. Now we've broken it down all the way down to CO2. We have broken down all of the bonds formed in glucose, and now we've created a lot of CO2. So that's when we respire. Oxygen goes in, and what comes out? CO2. So where does that CO2 come from? It comes from the oxidation of glucose. So in the citric acid cycle, glucose is completely oxidized to CO2. So glucose, the citric acid cycle, is located in the mitochondrial matrix. And the reactions start with the beginning of acetyl-CoA and with citric acid, and the last acid is oxaloacetate. Now the citric acid cycle has 8 organic acids. So a question is, do you need to know all 8 to understand cellular respiration? Well, maybe later on, but for this lecture, all you need to know is that this is a cycle and that the first acid is citric acid cycle, and the last organic acid is oxaloacetate. Okay, so what happens in this cycle? It says here 3 NADHs are reduced to NADH. 1 FAD is reduced to FADH2, and ADP or GDP is phosphorylated. Remember, phosphorylation is the movement of phosphate. So here is another example of substrate level phosphorylation, which occurs in step 1, which is glycolysis, and in step 3, the citric acid cycle. So remember, don't let GDP or GTP confuse you. Remember, ATP is an activated nucleotide, but there's 3 other activated nucleotides too. So it's not only ATP, but remember apple tree chewing gum. So here we have guanine, and it is activated and it's producing GTP. So it says here, the citric acid cycle turns twice for each glucose molecule. And remember at the very beginning, glucose in glycolysis is broken down to 2 pyruvate molecules, and that's why the cycle has to turn twice, one for each of the pyruvates. In this diagram here, you can see all of the organic acids, 1, 2, 3, 4, 5, 6, 7, and 8. the first organic acid is citrate, and the last one is oxaloacetate. So notice here we have acetyl-CoA, and notice you can see the two carbons here, 1 and 2. It's going in, and notice here it's broken down and we have a release of CO2. We have another release of CO2. And then notice with the breaking of the bonds, we have the release of electrons, which are accepted by NAD. NAD becomes reduced, and it becomes reduced here. And here we have energy that is being used to actually to reform or synthesize ATP. In other words, there's enough energy to reconnect this inorganic phosphate to ADP. When I say reconnect, the proper word is phosphorylate ADP, and that will make ATP. Or GDP to synthesize GTP. Notice there's some more electrons that are released, and FAD becomes reduced. And here you see NAD is reduced again. So here a lot of energy is released from breaking all of these bonds in acetyl-CoA. So here are the 8. Here are all of the 8 steps referring to the organic acids, and these are the enzymes that help catalyze the reaction. How is the citric acid cycle regulated? Notice feedback inhibition. So if you're asked how is cellular respiration regulated at each step, the answer is feedback inhibition. It also says reaction rates are high when ATP and NADH are scarce. That makes sense. When you need energy, then the system is going to be turned on. When you have a lot of energy, it's going to slow or stop the system. So here you see feedback inhibition. So if there's a lot of ATP, it's going to stop the citric acid cycle. Okay, so what happens to NADH and FADH2? So for each molecule of glucose, it says here glucose is oxidized. The cell produces 6 CO2s. That makes sense because glucose has 6 carbons, 10 NADHs, 2 FADH2s, and 4 ATPs. So what happens to the electron carriers? Remember, energy cannot be created nor destroyed. It can only be transferred or transformed. So where's all of this energy going in the form of these high energy electrons? Well, the energy is going to go to the last step. And so here you can see all of these electron carriers, step 1, step 2, and step 3. So where are all of these going? These are going to go to the very last step, step 4, the electron transport chain. Okay, so if I look at this graph here, it says the oxidation of glucose. And remember, isn't that what cellular respiration is? It is the oxidation of glucose. So the y-axis here says free energy change. So notice here, we're at the very, very top. Here, glucose has a lot of potential energy. But as we go through all of the steps, what's happening? Glucose is losing its energy. But remember, energy cannot be created nor destroyed. It can only be transferred or transformed. And that energy is being transformed to what? Either ATP or these electrons that are being carried by NAD or FADH2. And I'm sorry that the picture of myself is hiding the FADH2. Most glucose's original energy is contained in these electron carriers, these like little molecular taxis. So they have to go somewhere to leave off these electrons and protons or hydrogens. So how is it going to do so? So this is where oxygen comes in, and this is why we inhale air that contains, or the atmosphere that contains oxygen, because we need the oxygen to actually produce ATP in step 4, the electron transport chain. So it says here, NADH is oxidized when combined with the inner membrane of the mitochondria. But remember, what does oxidized mean? Are we losing or gaining electrons? Yeah, you're right, we're losing electrons. So remember, in prokaryotes, there is no mitochondria, so it's happening at the plasma membrane. Okay, so biologists have found that the membrane components cycle between oxidized and reduced state. In other words, there's a lot of transfer of electrons, and I always go from high to low energy, high to low energy. Okay, so here we have the electron transport chain. This is where all of the electrons will be deposited. So notice here that there's a lot of other molecules that are involved. Here we're talking about ubiquinone. We're also going to see that iron is involved. Boy, these look like coenzymes and cofactors. Okay, so I want you to hear about them, but you don't need to exactly know them for the exam. But what I do want you to know is that electrons are going through a series of redox reactions. They're going from high energy to low, high energy to low. And when my hands are shifting back and forth, there's a series of intermediate molecules that are helping with these redox reactions. So electrons are going to move from one molecule to another, and as they do that, they're going to start to lose energy. They're going to start to lose energy. They're going from a high energy to a low, high to low. Okay, so you know that there's cytochrome C and ubiquinone represented by Q. The other thing that I need to tell you is these molecules are located among 4 protein complexes, and these are given Roman numerals, and we're going to see these in an up and coming diagram. And as the electrons are released, these high energy electrons are released, this energy is going to be used to pump protons, hydrogens, on one side of the inner membrane of the mitochondria. Okay, so here we see these complexes in these like gray circles here. Here's complex 1, 2, 3, and 4. The other thing that you're going to see is NADH, notice they too have an initially high energy, but as the electrons go through a series of redox reactions, they're going to lose their energy. But where is this energy going? This energy will be used to pump protons on one side of the membrane. So notice here we have iron and sulfur and cytochrome C and ubiquinone. Now notice that this is a prosthetic group, and here is a coenzyme, and these are cofactors. I sometimes feel tired, and doctors will prescribe iron. So now you know why you need iron, because iron is used to help harvest energy in step 4, the electron transport chain. Okay, so these are all of the detailed steps needed to actually have these high energy electrons, that energy to be transferred to pump protons to one side of the membrane. Okay, so it says here, the role of the electron transport chain. The energy from the redox reaction is used to pump protons. And remember, protons is another word for hydrogen. And this will pump protons on one side of the inner membrane. So now all of that energy from glucose, and this is the majority of energy, will be used now to actually create this electrochemical gradient. I love this diagram here because look, at the very beginning we see NADH and FADH2. These are our electron carriers, what I've been referring to as little molecular taxis. And you know that they're reduced, but they're going to quickly become oxidized. In other words, they're going to let out their electrons. And notice here's complex 1, 2, 3, and 4. And when I look at these complexes that are embedded in the inner membrane of the mitochondria, they remind me of volcanoes. And the reason why I say that, the energy coming off from these electrons will be used to pump protons on one side of the membrane. So what's being pumped are these protons. So as the electrons move from one complex to another, they eventually lose their energy. So remember, we just can't dispose of the electrons. They've got to go somewhere. And notice that's where oxygen comes in. This is why we breathe in oxygen. And so all of the hydrogens and all of the electrons will bind with oxygen to eventually form water. So water is one of the products from the electron transport chain, ETC. Okay, so how is it that we're actually going to synthesize these ATP? Well, there's one other thing that's embedded in the inner membrane besides complexes 1, 2, 3, and 4. And the fifth thing that's embedded is ATP synthase. So what is ATP synthase? Well, if I look at the end here, ACE refers to enzyme, and synthase is saying it's synthesizing, it's making something. What is it making? It's making ATP. So this is acting like a turbine, like a motor, and as the hydrogens cascade down, they're going to turn the motor around, and this energy will be transferred to phosphorylate ADP and make ATP. Okay, so what is this process called? So we have this proton gradient, and this proton gradient is often referred to as chemiosmosis. Chemiosmosis. And as it moves through ATP synthase, because of this electrochemical gradient, as it moves, all of the protons on one side will come cascading down, and the only place they can cross the membrane is through ATP synthase. And as it does, this proton motive force, this energy, this motion is going to create ATP. Now, they did an experiment, and there is an experiment in the book that shows you how they figured this out, but it personally doesn't help me. So I have a hard time explaining it, but maybe to help yourselves, please read so in your book. So I'm going to gloss over this part. And here's a continuation of the experiment. Please read it in your Pearson book. Okay, so now let's get to this part. What this slide is telling you is that ATP synthase is an enzyme, but this enzyme is made of many parts. There's a knob, there's a base, there's a stator, there's a rotor. So this is a complex enzyme that is actually going to be used to harvest energy from this cascade of protons moving through from one side of the membrane to the other side within the mitochondria. You know what this reminds me of? It's like when you're reading those nursery rhymes and you see the old-fashioned water wheel by the river and water's coming through and the wheel starts spinning. This is the same thing, except what's driving the spinning motion of ATP synthase? It's actually the movement or the cascading of all of the protons. Okay, so here we see ATP synthase. Notice that it is embedded in the inner membrane of the mitochondria, and notice that it's complex. Here's the base, here's the F1 unit, here's the rotor, here's the stator. So you see that it's this complex enzyme. And then you see this electrochemical gradient. All the protons on one side, or all of the hydrogens on one side of the membrane. And the only way that they can come across is through, the only gateway is through ATP synthase. So as the protons start cascading down, then it's going to spin this F1 unit. And as it spins, look at what's going to happen. Inorganic phosphate is going to be reconnected to ADP, and that is going to form ATP. So the energy to produce ATP comes from a proton gradient. In other words, all of those cascading protons will spin that F1 unit, and that F1 unit is going to produce or find the energy or create the energy to synthesize ATP. So all 4 steps combined produce 29 ATPs. That's step 1, 3, and 4. Remember, pyruvate processing does not produce any ATPs. Step 1, glycolysis produces 2 ATPs. Step 3 produces another 2 ATPs. And the remaining 25 ATPs come from step 4, the electron transport chain, the step that requires oxygen, the step that requires oxygen to receive low energy electrons and protons that will also produce water. This is my most favorite figure in the whole chapter because it shows steps 1, 2, 3, and 4. It shows like where they happen. It shows what goes in, what goes out, what happens to the energy, and how might it be regulated. Well, we already know that all the steps are regulated by feedback inhibition. So this is my most favorite slide, and if you can go through and understand each of the steps, by golly, you understand cellular respiration. Okay, so this short video clip, you can find it in the other presentations, so you can watch this, and this is a great tool to help you understand cellular respiration. Now, the last thing I want to talk about is fermentation. So another way of describing cellular respiration is to call it aerobic respiration. Aerobic means that oxygen is present. So in the presence of oxygen, we're oxidizing glucose. Aerobic or cellular respiration. I can use those two terms interchangeably. Remember, so now cellular respiration, aerobic respiration, or the oxidation of glucose in my mind mean the same thing, and I can use all 3 of these interchangeably. Now, if there is no oxygen, then we can say it's anaerobic respiration. A or AN in front of a word often means no oxygen. So anaerobic, AN means no oxygen. So energy is formed from glucose in the absence of oxygen. So using oxygen is the final electron acceptor. Remember, all those cascading electrons and protons have to go somewhere. Oxygen is great because it's highly electronegative and it has low potential energy. So the distance between glucose and oxygen, there's a big gap and you can harvest a lot of energy. So that's the nice thing about aerobic respiration. But in fermentation, that means that there is no oxygen. So what is fermentation? There is no oxygen to accept all of those electrons or protons. So it ends up that fermentation is basically step 1. It is just glycolysis. So some form of pyruvate, as remember, pyruvate is a 3-carbon molecule, will accept the electrons from NADH. So glycolysis and pyruvate processing and the citric acid cycle, well, we don't have really pyruvate processing or the citric acid cycle in fermentation. So it is just simply glycolysis. So it says here, fermentation is a metabolic pathway that regenerates NAD from NADH. And so we have electrons, protons, and we have also 2 ATPs made by substrate level phosphorylation, and that is all that is made in fermentation. So fermentation is basically step 1, just glycolysis. I love figure 9.2 because it compares the 4 steps of cellular respiration or aerobic respiration with fermentation. And you'll notice that fermentation is basically just what? Glycolysis. It's only step 1. If you have oxygen in step 2, then you will go through steps 2, 3, and 4. So if oxygen is present here, then you have cellular respiration. If you have no oxygen while having pyruvate processing, then it stops. You just have glycolysis and you have fermentation. Now, we can have 2 products from fermentation. We can either have lactic acid or lactate, or we can have ethanol. Okay, so it says here, when our muscle cells cannot get enough oxygen, they shift to lactic acid fermentation. So when I don't exercise a lot, I start getting itchy when I exercise from like crunching a lot of, doing a lot of lifts with weights. Okay, I'm just joking. But I start getting itchy, or when I go on a walk, my legs will start to itch. What is that? I have used up all of the oxygen, and I'm actually now going into the production of ATP without oxygen. So my body is producing lactate. You know, when you exercise and they say feel the burn, what is the burn? The burn is lactate or lactic acid. So this shows you in this type of fermentation that glucose is broken down, 2 ATPs are made, and electrons are released. Of course electrons are released because we're breaking down glucose into 2 pyruvates. But where do those electrons go? They have to go somewhere. Well, pyruvate accepts those. They're an electron acceptor, and when they accept it, then the product will eventually be lactate. So here you see 2 lactates being formed. Now you can also have alcohol fermentation. You know, the alcohol that you see in adult beverages, this also comes from the breakdown of glucose in the absence of oxygen. So we can have that also being produced too. And some form of pyruvate will accept the electrons, and that pyruvate is converted into acetaldehyde and CO2, and eventually that will form ethanol. And then cells that form other types of fermentation are used to make soy sauce, tofu, yogurt, and cheese. So we use fermentation in industry all the time to produce a lot of food products. Okay, so here you see fermentation, and in this case, these electrons are being accepted by pyruvate, some form of pyruvate, that will eventually make ethanol. Fermentation is much less efficient than cellular respiration. I'm going to say that one more time because I know that that's a test question. Fermentation is much less efficient than cellular respiration. Fermentation only produces 2 ATPs, whereas cellular respiration produces 29 ATPs or more. Now, there are some organisms that can do both fermentation and cellular respiration, and these are called facultative anaerobes. And as a matter of fact, E. coli, which is found in your gut, is one such organism. And this is the end of cellular respiration and fermentation. Thank you so much.